So today we're going to be doing some of the practice problems from chapter 3, which was about nucleotides, nucleic acids, and genetic information. So hopefully you all watched the lectures, and now we'll go through those practice problems that were in the slides. So first we'll do the review problems from the structure and function part of the lecture. And first we'll discuss those nitrogenous bases. Can anybody tell me what are the 5 bases that are found in nucleotides? Yeah. Uh-huh. Oops. I'm listening. Uracil, adenine, guanine, thymine, and cytosine. And which one of those, besides thymine, which one of the other 4 pairs with thymine? Or more? What? Yes, adenine pairs with thymine. What else can pair with adenine? Uracil. Yeah, excellent. But thymine can only pair with adenine, and as we also know, guanine can only pair with cytosine. So can anybody tell me which ones of those are purines? Yeah. Exactly, yeah. Adenine and guanine. And the ones that are not purines, what are those called? Does anyone remember? Pyrimidines, yeah. Great. So those are what the bases themselves are called. And here are pictures of them all in case anyone needed a reminder. So as we mentioned, on the left we have our purines, adenine and guanine. On the right we have our pyrimidines, cytosine, uracil, and thymine. Which type of nucleic acid has thymine in it? Yeah, DNA, exactly. And which one has uracil in it? RNA. Yeah, great. So that is our bases. Great job. So now we have a molecule. It may have a familiar looking structure. This happens to be caffeine, which probably a lot of you are familiar with because it is in the coffee if you are a coffee drinker or a tea drinker. So if you're looking at caffeine, it has this ring structure to it. What does it remind you of? Does it look more like a purine or more like a pyrimidine? Yeah. So that's okay. So pyrimidines are the ones that have a single ring, right? They just have a single 6-membered ring. Purines have the double ring. It happens. It's fine. Yeah. So this one is actually a derivative of purine. So let's talk more about this molecule on this slide. So first, let's look at the sugar part of it. Does it have ribose or deoxyribose? Yeah. Great. And how do you know that? Yes. Yeah. So this carbon has an OH. That's the 2' carbon. It has an OH, so it's ribose. Deoxyribose, as you know from the name, doesn't have an oxygen in that position. It would just have an H there. So is ribose found in DNA, RNA, or both? Yes, it's only in RNA. And how do you know that?
Ribose nucleic acid?
Yeah, exactly right. It's in the name. So DNA is deoxyribonucleic acid. It comes from deoxyribose. RNA is ribonucleic acid. It's got ribose. So now let's take a look at the base on this molecule. Is it a purine or a pyrimidine? Yeah, it's a purine. Yeah, it's got that double ring structure. It's a purine. So when we consider this molecule as a whole molecule, the sugar and the base, is it a base of nucleoside or a nucleotide? Yeah. Yeah, it's a nucleoside. How do you know that? Exactly. Yeah, so a nucleoside has a base plus a sugar. A nucleotide has the base plus the sugar plus the phosphate. And as you correctly identified, there are no phosphates here. We just have our base and our sugar, so that makes it a nucleoside. So for the last part, can anybody name this molecule for me? I guess first of all, can anybody tell me which base it is? That might help us get started. Yeah, it's adenine. Yep. So knowing that it's adenine, what could you call the molecule? Yeah. Yeah, so this molecule is called adenosine. So the names from the nucleosides come from the names for the bases. So it has adenine in it. It's called, when you add a sugar on, it's adenosine. If it was different bases, the nucleosides would have other names. So like guanine, the nucleoside would be guanosine. So those are both purines, right? Adenine and guanine. When they become a nucleoside, you turn the end into sine for the purines. For the pyrimidines, the name is a little different. So cytosine is one of our pyrimidine bases. When that's a nucleoside, it's called cytidine. Thymine, the nucleoside is called thymidine. And uracil is called uridine. And then if we wanted to name it when it was a nucleotide, how would we do that? If we added on phosphates to there, what would we call this molecule if it also had phosphate groups here? Exactly, yeah. So if you had 3 phosphate groups here, you would call it adenosine triphosphate. So when we name the nucleotide, we use the name for the nucleoside and we add on how many phosphates are there. Does that make sense for everyone? Yeah. That's right. Yeah. So it's important because, yeah, the way we name this, it's for RNA, right? So, yeah, we actually wouldn't see thymidine biologically because that would imply that it has ribose, right? So if we actually, in DNA, you would see deoxythymidine. And it's the opposite for uridine, right? You only see uridine. You never see deoxyuridine because uracil isn't found in DNA. For all the other ones, you can have either one. So because this sugar is a ribose, we called it adenosine. If we took away this OH, it would be deoxyuridine. deoxyadenosine triphosphate. Or if there were phosphates here, we would call it deoxyadenosine triphosphate. Great question. Thank you. Any other questions about this? Yeah. I would say definitely no purines versus pyrimidines. Don't stress too much about recognizing like adenine versus guanine. I know. Sorry, I was just looking at the amino acids and trying to like bring my brain back to the DNA, what's on the exam. But yeah, don't worry about the specific structures. I'll give you them if you need them. Yeah, yeah. Great. Any other questions? Okay. So, does anyone want to be a brave volunteer and draw the appropriate hydrogen bonds between either one of these pairs of bases? Come on up.
Just the first one. Just draw the long pair.
No,
that's fine. Okay.
Just like where they would go. Thank you. So how many hydrogen bonds do we have between adenine and thymine? As Christine just drew here, we have 2, right? Does anybody want to draw the hydrogen bonds between guanine and cytosine? Go for it. Fabulous. Yeah. So guanine and cytosine have 3 hydrogen bonds between them. So we talked a lot about what conditions we need to have to make a hydrogen bond. So what are those conditions and are they being met here? So can anybody tell me one requirement to make a hydrogen bond? Yeah. Yeah, exactly. So your hydrogen has to be attached to a nitrogen, oxygen, or fluorine, and the hydrogen bond has to be made to a nitrogen, oxygen, or fluorine. What is the other rule for making a hydrogen bond? Yeah, it has to be linear, exactly. And that's exactly what we see in this hydrogen bonding between the bases, right? We have our hydrogen in the middle, it's covalently bonded to a nitrogen, and that makes this linear bond to an oxygen. And we see that for all of them. They're all linear, and we all have hydrogens attached to either a nitrogen or an oxygen, or I guess they're all attached to a nitrogen, and then they make covalent bonds they make hydrogen bonds either to a nitrogen or an oxygen. Any questions about the hydrogen bonds between the bases? All right. So here we have a polymer of nucleic acids. This is definitely a little bit busier looking than the one we were just looking at a couple slides ago. So we have 4 different nucleotides here, right? So first of all, can anyone tell me, is this DNA or RNA? Yeah. It is RNA. How do you know that? Exactly, yeah. So we have these hydroxyl groups on the 2' carbon. So we know that that gives us ribose. And we know that ribose gives us RNA. We can also see from the structure that we have our phosphate groups attached to the 5' end of all the ribose, all the sugars. So now we have the string. That's how we know we have a string of nucleotides, right? We have a base attached to a sugar attached to a phosphate. So that's one nucleotide. And then there are 4 of these in this molecule. And the way that it's usually conventionally drawn is with the phosphate on the 5' end, because that's sort of the way it gets built up in biological systems. So we usually will conventionally have the 5' end has the last phosphate, and the last 3' end on a polymer like this would have the OH. And then it's sort of ready to accept another phosphate from whatever the next nucleotide that you're going to add on as you build up this string of nucleotides. And the nucleotides generally get built up from starting from the 5' end, and then you add on to the 3' end as you go. So if we were to draw a complementary DNA strand, what would that be? So here I just have written out what we have drawn over here, which I've now covered in pen markings. So we have, at the top, we have adenine on the 5' end, and then uracil, cytosine, and guanine. So that's written out here, 5', AUCG, 3'. So how would I know what the complementary DNA strand would be for this sequence? What's complementary to A? Or does someone just want to tell me the whole complementary sequence?
5' to 3'.
Uh-huh. Yeah, exactly. So whenever we have a double strand, we have DNA that's complementary to RNA or DNA that's complementary to another strand of DNA. They're always anti-parallel. So in this case, I drew the top strand from 5' to 3' towards the right. So the complementary strand is going to go 5' to 3' towards the left. And from there, you can fill in the bases that are complementary to each other. So G matches with C. There's 3 hydrogen bonds. C matches with G again, 3 hydrogen bonds. The uracil will match with an adenine. And then since this is a DNA sequence, the A will then match with a thymine. Does that make sense to everyone? Okay. So if a piece of genomic DNA, that's double-stranded DNA, contains 30% A, what will be its percent content of G? I'll give everybody a couple minutes to do some math. All right, I see most people seem like they have put down their pencils, so can anybody tell me what is the percent content of G? Yeah, 20%. Yeah, and how did you figure that out? Yeah, exactly. So this is using Chargaff's rule. And that states that in any double-stranded nucleic acid, your percent A equals your percent T. Your percent C equals your percent G, because they always will hydrogen bond with each other. So when you have one of the other. And we also know that percentages always add to 100. So if we have 30% A, we know we also must have 30% T, 60% right there. So the other two must add up to 40%, right? Because that makes 100. So we can split 40 in half to figure out how much C and how much T. So half of 40 is 20 and 20. That make sense to everyone? Any questions about Chargaff rule? Awesome. So if I had a single-stranded piece of RNA, could I use this rule to figure out percentages of nucleotides? I'm seeing heads shaking no. Why not? Yeah. Yeah, so if you just have a single strand of a nucleic acid, you can have whatever percent of A, T, C, or G. They're not related to each other. They're only related to each other with this rule when you have a second strand that has the complementary pairing. So if you have an A on one strand, you have to have a T on the other strand, for example. But if you just have one strand, there's almost, I'm not going to say no rules, but there can be a large variability in how much you have of each nucleotide. Does that make sense to everyone? Any questions? All right. So let's talk about the central dogma of molecular biology. So we've talked about all of this before, maybe not exactly called it the central dogma of molecular biology, but this is the sort of fundamental idea behind how we think genetic information gets expressed and used in most living organisms. So everybody can just take a few minutes and see how many blanks they can fill in, and then we'll come back together and go over it together. All right, with that final sound, let's go over this slide. So genetic information is stored in the form of blank in the nucleus. So who can tell me the first blank? Yeah. DNA. Excellent. And for extra thinking, you can also think of this as it's also stored in the form of chromosomes. So chromosomes are made of DNA, and the sort of format they're in is chromosomes, right? It's all coiled together in a funny shape. That is a chromosome. So chromosomes are made from DNA and also some proteins that kind of help to hold it together in that tight shape. So now that we have our DNA that is stored in the nucleus, when a cell divides, how do we copy the DNA? What enzyme does that? Yeah. Yeah, exactly. DNA polymerase. And what is that process called when DNA gets copied? Yeah, replication. Excellent. So that's sort of the first part of the central dogma. You can sort of represent that by, you have DNA, and you want to replicate it. We'll often write that as a circle. That's replication. That's the first part of the central dogma. But then if we want to express that gene, DNA has to turn into a different type of molecule. What molecule is that? Yeah. I heard someone say, yeah, RNA. Thank you, everyone. Yeah, so we make RNA. And what enzyme helps us make RNA? Yeah. Yeah, exactly. And what is that process called? Yeah. Transcription. Great. So now that we've made our RNA in that middle step, we have to make another type of molecule to finally express our gene. What molecule is that? Yeah, protein. Excellent. So what enzyme helps us make protein out of RNA? Yeah, ribosomes. Great. And what is that process called? Yeah, translation. So yeah, you can sort of write it out in the form of a paragraph, or you can sort of draw it out in a little smaller diagram, however it helps you remember. But this concept of how DNA gets replicated and also how it turns into RNA and then protein is what we call the central dogma. It's really central to understanding what we've learned so far about molecular biology and how that all works. Yeah. No, there are different RNA polymerases, and different ones will make different types of RNA. Great question. Yeah. 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 Yeah, so for this particular example, we're saying RNA in general, but yeah, we really mean messenger RNA because messenger RNA is the one that gets made into protein. That's exactly right. Yep. And we will actually be going over that in the next slide. So everyone, if there are no more questions here, then we will talk a little bit more about the types of RNA. So thanks for leading us into that. So I guess everybody just take a few minutes, see if you can remember the three major types of RNA and a little bit about what the function is of each one of those. All right, looks like most of you have finished thinking. So can anybody tell me one major type of RNA? Yeah. Yeah. And what does transfer RNA do? Yeah, exactly. So it helps with translation. And it does this by physically carrying over the amino acid that you need to the ribosome with the growing protein in it, and it hands off the amino acid onto the growing protein. That's transfer RNA. Anybody name me another kind of RNA? Yeah. rRNA. Great. What does rRNA do? Yeah. So ribosomal RNA also helps with translation.
Exactly does the big polymorphism from RNA side? Like your DNA would, the polymerase would make the rRNA, but the rRNA wouldn't go into another ribosome to make protein for the ribosome, would it?
No. Yeah. So the proteins that code for the the genes that code for the proteins that make up the protein part of the ribosome are different. So rRNA and tRNA never get translated. Their function is as RNA. So they stay RNA forever. They never get translated. Yeah.
So rRNA and protein together make a ribosome.
Yeah, so ribosomes are like big globs with a bunch of proteins and a bunch of rRNAs that come together and help work and do their job together to make proteins.
Got it.
Yeah. So they're also made using RNA polymerase. They're coded there's code for them in the DNA in your nucleus, and then when you need to make more of them, you can use RNA polymerase to make RNA out of that DNA template, and then in the case of tRNA, it will fold into a particular shape that we saw in the lecture. And there are a bunch of enzymes that help, for example, to like attach the amino acid on to the tRNA. And then they kind of wait around until they see their chance and they hop into a ribosome and hand off the amino acid. And rRNA is similar, where there's a gene in the DNA that codes for the rRNA. RNA polymerase comes, transcribes that to RNA, and then when you make the other proteins that make up the ribosome, the rRNA comes together with those proteins to form a whole ribosome. Well, there's two subunits of a ribosome that then later come together when it's time to translate a protein, but you got the idea. Any other questions about these two types of RNA? Or you wanted to give the third type? Yeah. Dangerous. Yeah, so it's mRNA. So what does mRNA do? Yeah. So it brings genetic information out of the nucleus. It has it codes for a protein. So yeah, mRNA is kind of the bridge in between the nucleus where your DNA lives. It has the code for all the genes you want to express. Your RNA gets made in the nucleus, then it leaves, it goes into the cytosol, where it is then able to get translated. And it gets turned from a nucleic acid code into an actual protein that's made of amino acids that has some kind of job in your cell. And that's why the process is called translation when mRNA is turned into protein, because you're sort of translating this genetic code into a different type of molecule. Any questions? Yeah. All of them are kind of involved, right? So mRNA is the code. It gets kind of fed into a ribosome, almost like, this is probably not a great reference for you guys, but like in a cassette tape, there's like the tape that gets like fed through a little slot, which like you probably have not seen. But yeah, I'm trying to think of some of you. Excellent. Great. So yeah, mRNA kind of has the code that gets fed through a ribosome. The ribosome, part of its structure is the ribosomal RNA, the rRNA. And then as the mRNA is getting read by the ribosome, every three nucleotides in the mRNA is called a codon, and that is a piece of code that is specific for one type of amino acid. So the tRNA then brings over that matching amino acid and adds it on, and then the ribosome helps to move the amino acid onto a growing protein. So really all three of the types of RNA are kind of involved in making proteins. One is the code, and the other two kind of help with the process of translation. Does that help? Okay. Yeah. So all of them have to get transcribed, right? So when you start out, everything is, coded by DNA in your nucleus. So for the DNA to turn into any of these three types of RNA, they all have to get transcribed by RNA polymerase. Any other questions? Yeah. Uh-huh. Correct. Yeah. There are several types of polymerases. Off the top of my head, I don't want to tell you, because I'm not sure if I'd remember off the top of my head which one is which, but yes, there are several different types of polymerases, and they're also different between eukaryotes and prokaryotes, so if you're considering all of those, there's a whole lot of different polymerases. But I can look that up and get back to you if you're interested in which particular ones. Any other questions? Yeah.
RNA and rRNA, are they like decomposing anyway? That's orbit, or they're made and then they come.
That's actually a really good question. So yeah, I mean, certainly, I actually don't know specifically for tRNA and rRNA, but in general, yeah, I mean, your whole cells sort of function through like signaling cascades. So like if you don't have enough of one molecule, that will sort of inform another molecule. You ran out, and then that can potentially send something into the nucleus to ask the DNA polymerase to make more of that gene, sort of in like a very general way. Specifically for tRNA and rRNA, I don't actually know like the specific like pathway of information of like who tells who what to do. But yeah, I mean, certainly as you make proteins, you're going to run out of amino acids on those tRNAs. Those have to get refreshed. I think the tRNAs themselves do last a long time, and they can get sort of reused, right? So if they drop off an amino acid, the tRNA can get recycled. You can add a new amino acid onto it. But I mean, over the lifetime of the cell, they'd have to degrade at some rate. Yeah, I mean, same thing for the rRNA. So the rRNA is part of the ribosome. It doesn't get used up during translation. It's just part of the ribosome, but certainly there's some rate of like natural degradation that would happen, if that helps. Yeah. Any other questions? Okay. So now we can talk about our favorite virus. So this is a picture of SARS-CoV-2. This actually says COVID-19. COVID-19 is the disease. SARS-CoV-2 is the virus. My bad for not fixing that. So yeah, this is a picture of SARS-CoV-2. So SARS-CoV-2 is a type of virus that has a genome that's actually made of RNA. So a lot of times when we're thinking about, typical eukaryotic organisms, or prokaryotic organisms, you think about their genome as being coded by DNA that's, in the nucleus in the case of eukaryotes. But some viruses actually don't do this. They don't bother with DNA. Their genome is actually sort of already made into RNA. It's just ready to go and ready to get translated. And when the RNA replicates, when it duplicates itself, it needs a special type of RNA polymerase that's able to copy off of RNA as a template and make it into more RNA. So we as people, we don't have that in our bodies because we don't need to do that. We never make copies of RNA out of other RNA, right? When we make RNA, we're using DNA as a template. So the RNA templates, the RNA polymerases in our cells are only able to read DNA to make into RNA. So SARS-CoV-2 has a special type of RNA polymerase. And so when it brings that enzyme with it. It actually has a code for that enzyme. So it sort of hijacks the ribosomes in your cells to make the proteins that it wants. And it uses its own genome as an RNA template that can get fed into your own ribosomes. And that's us, that's the host. So we do the virus's job. The virus hijacks our own cells because it doesn't actually have the ability to translate its own genes. It just has the code. So for coronaviruses in general, there are 5 protein coding genes that they all have in common. And there are also 6 additional genes that SARS-CoV-2 has that are different from other coronaviruses. And as the virus replicates, it's going to gather mutations. So as the genome gets copied, there's going to naturally be some mistakes that get made. And those mistakes might end up making RNA that codes for a different for a different amino acid than was originally in that position. And so you end up with changes. And a lot of those changes happen in the spike protein of SARS-CoV-2, which you can see on the outside here. So the spike protein is the part of the virus that's required to infect someone. It's the part that will stick onto a host cell and allow the virus to sneak its way inside. And so they are also the part of the virus that your immune system recognizes a lot of the time. So a change in the spike protein might arise spontaneously, and if it helps a virus sort of hide from your immune system, that mutation, that spike protein with that particular mutation might become more dominant over other spike proteins that have a slightly different protein sequence. So over time, the virus will tend to build up mutations or changes in proteins that are on the surface that your immune system can kind of see. And that way it can try to hide from your immune system. I don't want to say it's trying to hide. It happens to make mutations, and some of them happen to help, and some of them don't. The virus isn't trying to do anything. It's just living its life. So the other common place where mutations occur is in the nucleocapsid protein, which is actually inside the virus. It sort of helps to protect and give shape to the RNA genome. And that is another protein in this virus that is really commonly recognized by your immune system. And so it's beneficial to the virus to potentially make mutations or changes in this other protein, this nucleocapsid protein, because some changes might help it hide from your immune system. So does anyone have questions about that? This is sort of as more of like a, this is interesting thing. I don't really have any questions for you about it, but does anyone have any questions like about this sort of idea of having an RNA genome or mutations that can help with immune evasion? Yeah.
On a vaccine for the flu,
Mm-hmm.
and it's supposed to be an RNA that has to be or something like that.
Okay.
It's supposed to be able to kind of work better at targeting a much wider range of strains.
Yeah. So I can talk a little bit about the universal flu vaccine. So hopefully this is interesting for everyone. But so flu, same idea. So the flu is another type of envelope virus. The inside looks a little bit different from SARS-CoV-2, but we're sort of interested in the outside of flu. So flu also has a protein on the outside that it's kind of like the spike protein, but flu has 2 different ones. It has H and it has N. So like you might have heard of flu as like H5N1, H1N1. It's named because they have different types of H's and N's. Different strains have different H's and N's. And so it's really hard to make a flu vaccine that helps your immune system recognize all the different types of flu because there are so many different H's and N's, and we don't know which ones are going to be sort of popular or circulating in one particular year. And the part of the H and the N that has a lot of variability to it is what we call the head domain, sort of the outside part that sticks out the most. There's, and this is especially for the H protein, but there's also the rest of it, right? So this part is called the head. The rest of it is called the stem. So the head has a lot of variability, just in general, like among strains of flu. The stem doesn't change a lot. So in different strains of flu, the stems are all very similar. So, and generally when you get infected with flu, your immune system is going to make antibodies against the head because that's the part that it sees. But if you get infected with a different strain of flu, the antibodies that you made against the first one you saw won't help you because they won't recognize it. If you can make antibodies against the stem, they might help you recognize a lot of different strains of flu. So the idea behind a universal flu vaccine is it's made to help you make an immune response that remembers what the stem looks like. So, I don't know, there's a lot going on. But yeah, I have my own opinions about whether it will work or not. But basically I don't know, I think it's too much to go into for this class. But yeah, so if you were going to make an RNA vaccine that was going to be sort of as a universal flu vaccine, you would figure out what part of the flu genome codes for making this protein that makes up the stem. Then you would make up some RNA. You'd protect it with some kind of envelope, like a lipid nanoparticle, for example, like the COVID vaccines, which you're probably familiar with, maybe. And then you could give someone the code to tell it how to make the stem protein. And then the cells in your body are like little stem factories, and your immune system will see all these stem proteins and make an immune response against them, and it will remember for, hopefully, when you get infected with the flu the next time, your immune system will say, oh, I remember you, you're bad. It will kill the flu virus and not allow it to replicate a lot or damage your lungs or any other part of your body. If that helps. That was a very long explanation. Do you want to know more about like RNA vaccines in particular, or like universal flu vaccines? Okay. That's a great question. And no, because RNA is actually, especially mRNA, is really unstable. If you just let it kind of sit around in water or in buffer, it'll just start degrading into nucleotides or like bits of nucleotides. It's not going to be one nice long strand that has your whole genetic code in it. So what we do to protect the RNA when we want to use it as a vaccine or as any kind of treatment is we protect it inside of an envelope. So kind of like we see on our virus, it has this RNA genome and it's protected inside an envelope. We use that sort of same idea. So if we have RNA for whatever gene we are trying to vaccinate against, we can put that inside of a lipid bilayer, and they're very tiny. They're called lipid nanoparticles. So the lipid nanoparticle is a lipid bilayer, similar to your cell membrane, right? Your cell membrane kind of protects the contents of your cell. Lipid nanoparticles are really tiny versions of this. They're sort of just big enough to contain some RNA, some mRNA. And that helps protect it. That and keeping it very cold helps protect it. And then when it's time to give it to a person, you let it warm up to be, room temperature, and you stick it in a person. And so the lipid nanoparticle helps protect the mRNA, and it also helps it sort of enter a cell. So because this cell membrane, because it resembles a cell membrane, it will help it kind of merge into an actual cell with an actual cell membrane. And there's a lot to go into for right now. This is more of like a cell biology thing. But so essentially then the mRNA can get delivered inside of your own cell, and then your own ribosomes will translate that RNA and make it into a protein. And that protein can get sent out of your cell, and then your immune system will see it and make a response to the protein in a very broad sense. Does that make sense? Okay. Any other questions about, viruses? Yeah. So you mean like the antibodies that we make and like the structure of the spike protein itself? Or okay. I would say they're fairly different. I know like in this picture it's sort of drawn as like a little Y shape with like a little ball inside, but if you actually look at a more detailed picture of the spike protein, it's actually like a trimer. It has like a very specific shape, and it has to do all sorts of things like get a cleavage in a certain spot, and its main goal is to basically recognize or get recognized by a receptor on your cells called the ACE2 receptor, and that's what helps the spike protein. That's how the spike protein tricks your cell to bringing the virus inside. It binds to this receptor on the outside of a cell, and your cell says, "Oh, I want some of this. Let me bring this in." The way antibodies work is pretty different. They stay outside of cells in the environment, and they're sort of patrolling in your blood and in your lymph to look for stuff that they recognize. But in general, I guess antibodies do also enter cells once they bind to an antigen and get recognized, but not. They use a different receptor. But I guess that sort of idea is the same. They both end up binding to receptors and getting pulled inside of cells, if that helps, but in a different way. Yeah. Yeah. So the nucleocapsid protein is pretty different. So on this diagram, it's written as nucleoprotein, but so the spike protein is on the outside surface of the virus. The nucleocapsid is on the inside. It's sort of integrated in with the genome. So it's sort of like how I was talking before about when your DNA is in chromosomes, it has proteins kind of mixed in with it that kind of help it keep its shape and keep it orderly. Those proteins in people are called histones. In the SARS-CoV-2 virus, for the RNA genome, it has these nucleocapsid proteins that have a similar function. They kind of help the RNA stay orderly and keep its shape. Yeah. Over here, B cells. Yeah, so B cells are one of the cells that are part of your immune system. They are the cells that make antibodies. So B cells are actually created through this process of like random addition and subtraction of like pieces of genes, and so your body makes a whole variety of B cells that can recognize almost anything. And so what your immune system does is it has all these B cells patrolling around that can recognize all different kinds of things. And when one of them does finally stick to something that it recognizes, it starts this whole cascade of an immune response to both help fight off the thing that it recognizes and also to create a memory for later in case you get infected again. So like that's sort of the principle behind vaccines. You inform your immune system. You're like, here's a thing. Do you recognize it? Your immune system recognizes it, and it makes a memory for later when you actually get infected with the actual pathogen. Any other questions? Okay, so let's go on to the last sort of line on this slide. So the way that the genes are coded in SARS-CoV-2 is some of them actually overlap with each other in the genetic code, and they're in different reading frames. And we'll talk about that a little bit more on the next slide. But I guess before we get into that, does anybody know what a reading frame is? Yeah. Yeah, exactly right. So let me try to make some space here on this slide. So anytime you have a strand of mRNA that has a code for something, so there's a bunch of bases, right? You might have A, U, G, C, whatever. This is just arbitrary. So you have this strand of RNA, and your ribosome is going to read them in groups of 3. So if you start at the beginning, you start by reading AUG, and then UCA, and then ACG. But you don't have to start at the beginning, right? You might start one base over, right? So you could also read this as UGU and CAA. Or if you slide one more base over, you could read it as GUC AAC. And those strings of 3 bases will each give you a different amino acid. So when you finally build up your protein, depending on where you started reading, you could end up with completely different proteins that you made out of the same original strand of RNA. Yeah. Yeah, exactly. So there are different clues that your ribosome uses to help it know where to start. So in this case, yeah, you would start from the AUG. But when you have genes that are coded in an overlapping way, there are multiple AUGs, and each AUG is in a different reading frame. So like if I were to make a better example for you, I could have AUG, C, AUG, A, G, G, U, C, for example. So if you were to start from your first reading frame, you would have AUG, CAU, GAA, GGU. If you were going to start, and then you would start translating from the beginning, from the AUG. If you start from the second one, you'd have UGC, AUG, AAG, GUC. So then your ribosome could say, oh, I'll start here at this AUG. Then you have a different set of amino acids. So yeah, so in this case, if you start here, you wouldn't end up translating those because you're going, this way. That kind of makes sense for everyone. Yeah.
So just to clarify, start at the start codon, or it doesn't matter?
Yeah, so in general, the reading frame is sort of like where you start from. So there are 3 possible reading frames, right? If you start from number 1, number 2, or number 3. So like if you start from the first reading frame, you could have a 4th reading frame, but really that's just the first reading frame again, right? Because that's the second set of 3 from your first reading frame. Does that make sense? So like there are 3 potential reading frames. You might not be able to translate from all 3. It depends on what the actual code is and if it tells you you're allowed to translate from there. Like if it has an AUG, like the start codon, which we will get into this in later lectures when we talk more about proteins. But yeah, so that is, yeah. Yes, yeah. So this particular code has 2 possible reading frames, the number 1 and number 2. Yeah, and there is no third. You can't make a protein out of the 3rd reading frame in this one because there's no start codon. In this particular one, yeah. So yeah, and so but when it's handy to do this to have overlapping genes because it saves you space, right? It's kind of like, I don't know, compressing a file or something. So you have a relatively small amount of RNA that can give you code for a whole bunch of different genes. And so that's what a lot of viruses take advantage of, especially because viruses are very small. They have a very limited amount of space to stick code into. And sort of the more space they can conserve, the more efficient they can be, like when they're replicating and everything else. Yeah. Yeah. That's a good question. I believe they are in both, but it's been a little while since I thought about that. I'll get back to you to tell you for sure. Yeah. Any other questions before we talk more about reading frames? Okay. So the SARS-CoV-2 genome, really should have proofread this. Yeah, so it's made of RNA, and this is sort of a nice little picture that was borrowed from the New York Times that has different colors that sort of represent the different proteins you can make. So I know it's kind of small and hard to read, but hopefully on your actual slide you can read it. But over here in this light yellow, you have the ORF1A, so that refers to the first reading frame. So the ORF stands for open reading frame. And so this ORF1A protein has a number of different proteins that it gets broken down into. So something that's sort of interesting about this virus and other viruses. So compared to people, when we want to make a protein, we have one piece of RNA, and that one piece of RNA generally gets made into one protein. Some viruses, they'll actually make a really, really long piece of RNA that has amino acid codes for a bunch of different proteins. And those proteins kind of all get made all together, and then they get chopped up at the end into their separate proteins. So they make what's called a polyprotein. And to cut up the polyprotein into the individual proteins, it also needs to use proteases. So that's why this diagram is sort of labeled. You have this ORF1A protein. It makes a very long protein that actually gets cut up into all these individual proteins, all these non-structural proteins that are labeled NSP, and then all these other labeled proteins, 5, 6, 7, 8, 9, 10. And then going sort of around on the genome, there's another big long protein that gets made from ORF1B. That's a different reading frame. So we have reading frame 1A, and reading frame 1B, and they make a completely different set of proteins. So ORF1B gives you another non-structural protein, or a bunch more non-structural proteins. And then finally, at the end here, in the other colors that are not yellow, we have our structural proteins. So the big structural protein that you're probably familiar with is the spike protein, that's shown in red. And in this picture in the middle, it sort of shows you where it goes, right? So like this red is the spike protein that goes on the outside. And then you have these other proteins and other reading frames. You have ORF3A, you have the M protein, that's part of the membrane. You have the N protein, that's the nucleocapsid, that stays on the inside. So, I mean, all that is to say, don't worry about like knowing all of these different genes and proteins. Just sort of be aware that viruses have some of these special, interesting features, like being able to read out of different reading frames to code for a wider variety of proteins than you might otherwise think is possible. And yeah, I think that's pretty much everything I wanted to say about SARS-CoV-2 for now. Does anyone have any questions about any other of this stuff that we were just talking about? Yeah. Okay.
I have a really quick question.
Sure.
So is it an RNA? How does it replicate its nucleic, its genetic information?
Yeah, that's a great question. Yeah, so it has, I think I mentioned this before, but it has a special RNA polymerase. So part of one of these genes, the non-structural proteins, is it's an RNA dependent RNA polymerase. So this is an RNA polymerase that reads out of RNA. So part of the genome codes for this RNA polymerase.
Yeah.
Once the virus enters your cells, it uses your ribosomes to translate that polymerase. And then once your cells made the polymerase, that polymerase is going to go find the viral RNA and replicate the viral RNA. And the more it gets replicated, the more it gets translated, the more it can replicate until your cell bursts. Yeah. Any other questions? All right. So this is just sort of a little bit more about SARS-CoV-2. So we, as you're probably aware, over the last several years, there have been these waves of different variants that have arisen. And that kind of comes about from what we were talking about before. So the spike protein might gather a couple of mutations in the genome that make the spike protein look a little bit different, and that might help it hide from your immune system, or it might make the virus easier to pass on to your friend next to you, or some other reason that would make that particular mutated version of the virus more easily able to replicate and spread to other people. And so that's sort of how these waves arise. And so over time, like one will dominate, and then maybe some more mutations will occur in that, and then we'll move on to the next wave. And then once a lot of people have been infected with that, it gets passed around. There's a lot of mutations that build up, and then the next one arises. So each one can sort of grow as it, either because it's more contagious or because it's able to hide from your immune system a little bit, and so it's able to replicate and spread. And does anyone know why we call them variants and not like some other word? Like I know before someone mentioned strains, right? When we talk about flu, we talk about strains of flu. So that terminology is also maybe a little bit, not controversial, but there's sort of different people use them in different ways. But essentially, within a species, if you have variability that makes the individual members of a species a little bit different, but not too different, you may call it a variant. There's just like some small like individual differences, like between, all of you in this classroom, right? You're all the same species of human, but you're each individuals. So you have maybe different patterns of gene expression, or your genetic code is slightly different from each other, but you're still all humans. So maybe you'd call each other variants on humans, right? And then for when we call something a strain, that actually usually refers to our immune response to it. So if one strain might, like we were talking about with flu, so if you're infected with one strain of flu, you make an immune response to it, you can recognize and remember that strain of flu. But other strains of flu are genetically and in appearance different enough that your immune system might not recognize them. So that's what makes it a different strain. So the difference between strains and variants is sort of really a little bit hard to define. Usually a strain is a larger difference than a variant, but there's sort of a spectrum in between them. So just one more slide about this. So this is just another sort of way of showing just the different percentages of the different variants that we've seen. So it's just another way of showing how over time, like as one might predominate at first, but then over time, like another one will replace it. So like this red one, like was really popular in, May of last year, but, within a couple of months, it had already been sort of fewer and fewer infections with this variant and more infections with the BA5 variant that eventually has come to predominate. And if anyone's interested, there's definitely more data about variants and infection on the CDC's website and in other places for sure. It's a fascinating topic, but yeah, there's it all comes from that RNA genome, and we're talking about nucleotides. So so do we have any questions about sort of nucleic acid structure, function, any of the stuff we've discussed so far? All right, so let's continue on with the nucleic acid techniques. Sure.
So just while we're on the topic, DNA viruses, they all have to use like reverse transcriptase to make mRNA.
No, so when you're going from DNA to RNA, that's just regular transcription, right? You just use a regular RNA polymerase. If you want to go backwards from RNA back to DNA, that's when you need a reverse transcriptase. So there are some viruses that have an RNA genome, but in the course of their life cycle, they need to get made back into DNA to then get transcribed and translated, which is... So yeah, so like if the virus wants to hide in your genome, it has to turn into DNA. So then you'd need to have that RNA turn back into DNA, insert into your genome, so then it can exist along with the rest of your genes. And that reverse process of going backwards from RNA to DNA is sort of a violation of that central dogma that we were talking about before. So like sorry about the slides. Yeah, so our central dogma tells us everything is moving in this direction, right? RNA is turning into DNA, is turning into protein. Other way around. DNA is turning into RNA, is turning into protein. So if you wanted to go backwards, it's breaking the rules. It's unusual. But you're right, that some viruses do that. Cool. Any other questions? All right. So, nucleic acid techniques. So, we're going to talk a little bit about PCR. So, I will give everybody maybe 5 or 10 minutes to look through these questions, and then we'll talk about them together as a group. All right, so let's wrap up with this question. So start with the first part. So can anybody tell me what is the goal of PCR? Yeah. Yeah, exactly.
I have a question on that.
Yeah.
How does it work with Sept?
Yeah, exactly. So I just can't write and talk at the same time. 1 second. So yeah, so that's why you have two primers. You have your forward primer that starts, kind of marks where you want to start. Your reverse primer marks where you want to end. Those obviously start on two opposite strands of your double-stranded DNA. And so in your first DNA cycle, those will go on for sort of as long as you let them. So usually you're doing this in a machine that has timing for temperatures, right? So it'll extend on as long as you're on your extension step for, 2 minutes or whatever. But then on your next set of cycles, when you have more primers that are annealing to those strands, then so your strand that started with your forward primer and kept going for a while, now when you separate that out and you start a new cycle, the reverse primer is going to stick onto that, be complementary to that strand on the other end, and then it'll stop where that strand stopped, which is at your forward primer, if that kind of makes sense. So for each subsequent cycle, almost all of the actual DNA copies that you make will just go between the two primers, if that helps. It's kind of weird to visualize, and it's a lot of drawing. If anyone, yeah. It doesn't. I mean, essentially, it's just it's a numbers game, right? So after a few cycles, you'll have so many more of the exact length ones compared to your initial set of copies. They're still there. You just kind of don't really see them because there's so few of them in comparison. Yeah. Yeah, so that being said, yeah, so PCR, the goal is to amplify a DNA template. You might be, studying, so maybe to study how a gene works, or maybe you want to make a protein out of it. Has anyone in here used PCR ever? Yeah. What did you use it for? Yeah, that's a great perfect use of PCR diagnostic. And there's a question about that a little later, so maybe you can chime in more about that when we get to that one. Does anyone else here use PCR for anything? Yeah. Very cool. Yeah. Presence or absence of gene. Yeah, so these are all great uses for PCR. Cool. So can anybody tell me some reagents that you might need to do PCR? Yeah. Uh-huh. Yes. Mm-hmm. What else do we need? Yeah. We need template. Yep. What else do we need? Yeah. Primers. Yeah, very important. Otherwise, the polymerase won't know where to go. Yeah, so those are the four main reagents that you need. Why do we use a Taq polymerase and not any other polymerase? Yeah. Yeah, Yeah, you could say it's like heat stable. So yeah, it keeps its shape and is able to still function even when it gets very hot. Because part of your PCR cycle is you start out by making it very, very hot to separate out your template strands. And if you do that and then you destroy your polymerase, then nothing else will happen. So very important to use a heat stable polymerase.
Here's a quick question.
Yeah.
Why can't you just denature and let it cool down and then add polymerase?
You could, but that's a lot of work. Yeah, I mean, most of the time when you set up a PCR reaction, you kind of set it all up and then you stick it in a machine that cycles through the temperatures for you.
Okay.
So it's like a set it and forget it, or what you will. So you could, after each cycle, pull it out, add fresh polymerase and let it go. But yeah, mostly that would be a lot of work. Yeah. Any other questions? All right. So can anybody tell me the main steps in a PCR cycle? Yeah. Sorry, go ahead. Yeah, so the first part is denaturing. Yep. So that's separating the template strands. What is the next step in a PCR cycle? Yeah. Annealing. Yeah, so what's annealing? So what's happening when we're annealing? What's annealing? Exactly, yeah. Then what is the next step? Yeah. Yeah, elongation or extension. And what's happening during elongation/extension? Yeah, exactly. So we have our DNA polymerase, our Taq polymerase. So that's when your DNA polymerase reads your template and adds on nucleotides to make the complementary strand. Great. So does anybody know how many copies of the DNA sequence that we'll have after 10 PCR cycles? Yeah. Close. Any other guesses or educated guesses? Yeah. Yeah. Yep. So for every cycle, we double what we started with, right? So if we start out with one DNA strand, we can multiply that by 2 to the power of however many cycles. So if you started out with one single copy of DNA, so if you have like one copy of your DNA, and then you did 2 to the 10th power, it would give you 1,024 copies of that one piece of DNA. Because every cycle we double, we just have 2 to the power of however many cycles we have. So we're getting close to the time, so I think we'll pause there for now. We'll pick this up on Monday because today's Thursday. And just general reminders for everyone. Quiz 2 is due tonight, so if you haven't done the quiz yet, make sure you get that in before tonight. And yeah, so next week we'll finish up chapter 3 and we'll get started on chapter 4. And if anyone has questions about anything, I have office hours after this. Have a great weekend.